had. For example, I had a genetic test from your sibling or from your father or from your mother who had similar symptoms, I might be appropriate for me to actually just do individual genetic testing. Typically, I would say that's not what we do, but there could be a situation where that would be appropriate. What we typically do is panel-based gene testing, and so these are genetic testing to look for all known or likely LGMD genes or myopathies. What's important, and what we just heard about, is different panels and different genetic test results. Just because you had genetic testing doesn't mean that you had all of the same genes done, and we keep adding genes and removing genes from gene panels. And so just because you had genetic testing might not mean that you had everything done. And so knowing who did your genetic test result or who did your genetic testing, which company, is really helpful, because different panels exist for different genetic test companies. Another thing that's important, and patients will come to me and say, oh, I've already had genetic testing, and I'll find out that they had genetic testing maybe 15 years ago. And they may not have a diagnosis, but they had old genetic testing, and we've updated genetic test results, and we've added new genes. And so making sure that you've had updated. Now, if you already have a diagnosis, I'm not talking about that. I'm talking about if you don't have a diagnosis. And finally, we can do things where we can actually interpret all of the coding genes. And so this is something called whole exome sequencing. Again, knowing the genetic test that you had, so just saying you had genetic testing might not mean that you had everything done. And again, this is if you don't have a diagnosis, because even whole exome sequencing can miss certain things, deletions, non-coding regions. And so someone may say, well, I've had every gene tested. Well, that may be true, but I don't know what that actually looked like and what that genetic testing, what it was. So that's just important to do. And then later we can do something called whole genome sequencing. I want to talk quickly just about some of the barriers to genetic testing. And so currently, free genetic testing occurs quite easily, point of care in the clinic. But not everybody has access to that either. And whenever I say free genetic testing, it's not really free, it's sponsored genetic testing. And so a genetic test company is usually aligned with some pharmaceutical company that's interested in identifying their disease gene. And so they've helped kind of improve the diagnostics. But there are barriers, and one is sometimes it can be cost prohibitive, so payers, so even for genome sequencing, it can be a payer thing. Patient knowledge, community knowledge. I would say one of the biggest barriers we have is physician knowledge. And so not all of us understand genetic test results and genetic testing. We need to partner with a genetic counselor. But I would say not everybody has access to a genetic counselor as well. And so I think physician knowledge is really important. And I would say, and this needs to change, but the original patient care guidelines, this is from the American Academy of Neurology. This is from over a decade ago. But the care guidelines then actually said that you should actually sequence the gene that you think is the gene that is causative, meaning that I'm supposed to know whenever you come in that you have a dysferlin gene because I'm able to kind of figure it out. And that level C evidence, and level C evidence is just my opinion. It's Peter's opinion. It's our expert opinion. Suggests that we should do panel-based sequencing, which is what we do. And so this all changed, I would say, for limb-girdle muscular dystrophy in particular a decade ago. And this was free genetic testing that was allowed, point of care. And so point of care genetic testing is important because if I see you in clinic and you've come from far away, and if I'm able to do genetic testing that day in clinic, that's really valuable, because often what we would do 10 years ago is people would leave, I'd contact insurance company, we'd get lost to follow-up. And so by doing point-of-care genetic testing, we were really able to break down some of these testing barriers. And this was funded by the MDA as well as the Jane Foundation. And this here is the result. This kind of pie chart here kind of shows what they discovered. And they discovered that the prevalence of limb-girdle 2A was much more common than some of the others, and this was really, really helpful. They only sequenced 35 genes, But one thing that happened is although this was the result, what really was the result was that about 75% of people were left without a diagnosis. Half of them had what we call this variant of unknown significance that we just heard about, a VUS. And I would say that a variant of unknown significance leads to really a non-diagnosis in many situations. And so half of patients had variants of unknown significance in a limb-girdle muscular dystrophy gene. And so this leads then to the interpretation of genetic test results. We heard a little bit about it. This is a patient's genetic test result right here. And what you can see is that this patient has several different variants of unknown significance. This patient had a panel of 100 genes, yet they have variants of unknown significance in several muscle disease genes. And this is quite common. And I'm going to explain why this patient would have so many variants of unknown significance. And it really has to do with understanding human genetic variation. And so it wasn't until we actually sequenced the entire human genome, and this was obviously done 2 decades ago now. And what we learned with this paper right here, this was the first attempt at sequencing 1,000 patients and looking at how variable we are from human to human to human, how different I am from my genetics from one individual. And what we identified is that there is a huge amount of rare variants. And in fact, if we look here, many of us have common variants, but everyone has about 30 to 40,000 rare variants. And these are variants, in some cases, that are unique to you. And so when I say unique to you, that means unique to you. You have your own personal mutation. You've got it from mom or dad, so it's not entirely unique to you, but in many cases, it's unique to you. And so of these rare variants, 250 to 300 cause actual would be damaging to that protein. And in many cases, it can be on both alleles, suggesting that we are all have mutations that could be considered disease-causing, yet we're not all walking around with different diseases. And so the other thing that's known is that in many cases, all of us have between 0 and 7 pathogenic variants. So all of us have variants of unknown significance. And so now whenever we look at this genetic test report, we can kind of understand there's so much genetic variability from patient to patient. I'm now sequencing 100 genes. Of course I'm going to find variants of unknown significance that are unique to individuals and that are then hard to understand. And so how do we interpret these genetic test results? And so I'm going to go back to kind of elementary school. I think they're teaching genetics now in elementary school. Elementary school genetics, where we have a DNA that's transcribed to RNA that then makes a protein. And the way we can think about it is the DNA is the cookbook, the RNA are the different recipes, and then we can say that ultimately the protein is the cake, okay? So that's what the product is. So we translate our RNA into a protein, and that protein is then, we'll say, a cake. And what I can say is that cakes can come in many different flavors, okay? And so I think that's what genetic variation is. We need to be thinking about the cake coming in very different flavors. And so what I want to do is to kind of walk you through how we would do interpretation of a genetic variant. And I want you to think of this recipe here as the genetic code, all right? And so that's what the recipe is. It's different protein amino acids, and there's different steps to making that chocolate cake. And so what you can see here is the recipe is now right there, and whenever I make this recipe, I'm going to get a chocolate cake. But what I am very clear of is if I delete half of that recipe, I'm pretty confident I'm not going to get a chocolate cake, okay? I'm pretty confident I'm not going to get one. And so that's where I can do variant resolution and say if the protein is truncated, I'm not going to get that protein. That means that protein's not being made. But sometimes it's a little more complicated. And so one example is here. If I don't preheat the oven, okay? I've cooked a cake and I've not preheated the oven. I've actually gotten a cake. And so that we could say, I can take a pretty good guess and say that's probably a benign variation, okay? okay? So I can look at that mutation and say, I think because of my experience, because of the way I've looked at other proteins, I can say that if I don't preheat the oven, I'm going to get a cake. But what about the example of where we leave out something like the baking soda, or we leave out this teaspoon of salt that we seem to need to add? I actually don't know if I'm going to get a cake. I might get a different cake. I might get a cake that doesn't taste good. And so those are variants of unknown significance. And what that means is I don't know what that change is going to do to the protein, and so I leave that as a variant of unknown significance. And so what are the challenges of variants of unknown significance? Well, they leave the patient with diagnostic uncertainty. They leave it an inability for our genetic counselors to do their job and to help with family planning because we just don't have a good sense. They leave the physician who's just as frustrated as the patient with diagnostic uncertainty. They can lead to inappropriate treatment and testing. They can lead to increasing healthcare costs and a whole variety of other things. And the other thing I would say that's really important is that because of all these variants of unknown significance, we start to not really know the true prevalence of many of these limb-girdle muscular dystrophy subtypes. And so I think that's an important aspect. And finally, the tragedy would be that you would miss out on some type of gene-specific therapy because we didn't actually have a diagnosis. And so again, the importance is clear. And I would say by resolving a variant, we help improve it for other patients' care. And so this again is an example of a genetic test result where this is a patient has progressive weakness and they've got several variants. And are these pathogenic VUSs or are these variants that are incidental? And so if we looked even through other genetic test results, everybody has variants of unknown significance, and everyone might have variants of unknown significance in multiple LGMD genes. And that's just because of our genetic variability. And so it leads us as clinicians to start thinking about how we're going to do this. We're going to do gene first, or are we going to examine the patient first? And just the ease and low-cost genetic testing, invariably patients are getting genetic testing done first. And I would say I've had patients that show up with a genetic test result that they got even though they don't have any clinical symptoms. And that's a really now challenging thing for me to understand is, does somebody actually have the disease whenever they're not manifesting it? And so we really need to work together. Geneticists and clinicians need to work together. And I bring this up, do we order the gene test first or do we examine the patient first? And so this is an example of a genetic test result. This is an autosomal dominant disorder. And in this case, this patient had a mutation in DNA JB4. It causes a limb-girdle muscular dystrophy. It says pathogenic. That's a slam dunk. We've done that. That's a diagnosis of this patient. But here's another example. This is a patient that came to me, handed me their genetic test report, and said, I have limb-girdle muscular dystrophy. They didn't have limb-girdle muscular dystrophy, but they had a genetic test report that was very ambiguous and very confusing to them because it said a variant of unknown significance in that same gene. And so what I want to point out is that a geneticist can tell you if it's rare, they can offer predictions, but they don't know if it's actually pathogenic in the patient. And that's where we need to partner with clinicians as well. And so again, here's just another example of some genetic test reports that are ambiguous. This is a patient that has a pathogenic variant in one, but a VUS in another. Here's another. Everybody has variants of unknown significance. And so I want to give you an example of what we would do to help resolve a variant of unknown significance if I was a clinician, or as a clinician. And so this patient has a pathogenic variant in calpain-3. He has to have 2 mutations. We talked about that. They have to have 2 variants. And so the other variant has a VUS. And so this is a 34-year-old man with limb-girdle muscular dystrophy unknown etiology. He's got several variants of unknown significance. He's got one in dysferlin. He's got 2 of them in, he's got 1 variant in calpain-3, and he's got a pathogenic variant. And what we would do is we would kind of walk through this patient. We'd say, well, what is his history? He developed weakness at the age of 25 with climbing steps. That tells me perhaps this is limb-girdle. He doesn't have a family history. He's got proximal weakness. He's got some features that might be specific for calpain-3. He's got some scapular winging. We then do other tests. So we would look and check his muscle enzyme and do electrodiagnostics. We likely would need to do a muscle biopsy. Not everybody needs a muscle biopsy, but in a situation where we have a variant of unknown significance, it may be prudent to do a muscle biopsy in order to make sure that we can understand this. And so this patient has an elevated muscle enzyme. He's got, we didn't do an MRI of his muscle, but you could do something like that. He had a muscle biopsy that actually showed that he had a muscular dystrophy, and he actually had some other studies done. So then we go and we look in databases, and we can see that this calpain-3 variant is unreported in population databases, that it may change splicing. And we go further and we try to understand, do the variants segregate? This is where we would get DNA from mom or dad or from siblings. And so in this case, we were able to get DNA from his mother and get DNA from his two siblings, and we were able to see that this was on 2 different chromosomes. We did other testing on the muscle biopsy that needed to be sent to a specialty lab that showed that there's a reduction in certain proteins, and then we actually did a Western blot to actually look and show that calpain-3 is absent. So my point is that variant resolution is complicated. It takes a lot of work. And so it requires a specialist that understands hereditary muscle disease, and it's time consuming. And I guess the complicated becomes more complicated because some of these disease genes we're finding actually can be inherited in different fashions, in dominant and recessive ways. And so it just becomes more and more complicated. And so how can we kind of solve this? How can we kind of solve this, this process? And this is where we started a program. We're working with a group called ClinGen. ClinGen is an NIH-supported group. They answer 3 important questions. One is this gene actually causative for a disease? Is a variant, so a VUS, is it actually associated with a disease? Is it pathogenic? And finally, if we do find these variants, is it clinically actionable? And so the way we do this, we just heard briefly, we aggregate all of this information together, we add it together, we kind of stack it together until we get levels of evidence. And so what I would say is that we need to do this and create these rules specifically for limb-girdle muscular dystrophy because there are specific things that we can do. And so one thing that we're working on, and am I running out of time?
Yeah, we're way over.
I'm way over. Okay, I'm going to go really.
Not just you, the whole stuff.
Oh, okay. It's not just me. So what I would say is at this point we have an incomplete library of genetic variants. And what we want to do is we want to create a complete library so that whenever someone comes in with their genetic test result, it says pathogenic, it says benign, so that whenever we do genetic testing on children before they get any disease, we can say whether they're going to have this disease. And so we've been working hard to kind of create these mutational libraries and do these types of scans, and there's other ways that people are doing it as well, using bioinformatics and deep learning approaches. And the goal will be to take this incomplete library and create a complete library. And so the question would be, well, where is this library being housed? And so this library is housed in something called ClinVar. And so ClinVar is a database that's open access that you can look at, and you can look at your variant and you can see if that variant is present. And so in working with Sarepta, with the Jane Foundation, who's helped fund some of this, we've been able to actually go in and actually have an expert panel look, and we can actually demonstrate that whenever you look in ClinVar, these things become likely pathogenic if we identify them as pathogenic, and they'll say FDA recognized. And this can only be done through an expert panel that we're working on. And I would say that we've had great luck in doing that. And this is just an example. I have a poster over here to kind of talk about our success. And so at present, I would say genetic test interpretation and what I call armchair variant resolution requires knowledge of both clinical and genetic muscle disease. And so that needs to be done by specialists. Systematic disease variant resolution algorithms that allow us to do that, and online deposition of these databases will help facilitate clinical care and and that we probably still need to be doing some enhanced clinical phenotyping where we examine the patient and just make sure everything fits together. And so, so I'm going to leave you with that. We actually have people that we have help with curating. We actually have patients that come on and help with curating, and I encourage you to reach out to me if you're interested in kind of doing that. So thank you.
Thanks, Chris. Okay, so our last speaker, before we get to the interview section, is Dr. Peter Kang. He's a pediatric neurologist, neuromuscular neurologist, and director of the Greg Marzoff Jr. Muscular Dystrophy Center and professor of neurology at the University of Minnesota. And he'll be talking about how do we discover new genes and advanced sequencing technologies.
Thank you. Many thanks to all the conference organizers for the invitation to speak. So let's go ahead and get started and talk about some new genes. These are my disclosures. And this is the outline of what we'll talk about. We'll go through some historical views, a recent example of a new gene discovery, and some new sequencing technologies, just to give you an idea of what happens when you participate in research studies for genetics. So going back in history, in the 1970s, DNA sequencing became more of a feasible thing for single genes or parts of genes. And this is now called Sanger sequencing because there are other technologies now available. And then PCR was discovered in the 1980s, and these were both Nobel Prize winning discoveries, so they were quite remarkable and helped us accelerate genetic discovery. And then the first muscular dystrophy gene discovered was the dystrophin gene in 1986, with the accompanying protein described in 1987. So what's happened since then is we've gotten a much better idea of what the genes and proteins are that are involved in muscular dystrophy. And there's an enormous list now of limb-girdle muscular dystrophy genes. This is a chronological list arranged by year of discovery, and you can see that alpha-sarcoglycan and calpain are the two first ones. And you'll see many of these. For those in the audience who have genetic diagnoses, the genes that are relevant are probably on this list. And the techniques we're using have become more and more sophisticated. I'll just illustrate with what happened with calpain and the discovery of calpain, because it was a lot harder back then. Not that it's easy now, but there are different challenges now. But there are 4 key publications that I could find. One was from the early 90s, where they found that the gene for calpain was on chromosome 15, and just finding the chromosome was a big deal back then, and this is before the human genome was sequenced, and so the human genome made a big difference. And then in a second paper, they narrowed it down to a specific section of chromosome 15, and then there was another paper where they narrowed it down even more. And then finally, in 1995, they hit on the calpain gene, and that has led to a lot of discoveries since then about calpain and its protein product. And so it was quite a laborious process, taking many years, and it's become quicker in large part thanks to the Human Genome Project. One thing that makes it difficult now is that the more recent genes are a little bit more rare on the whole, so that it's harder to figure out if a variation we find is truly disease-causing, and can we really convince ourselves and other people that it's a limb-girdle gene. And so I'll just mention a recent discovery. There have been a number of them, but this is one that we published a few years ago, a new LGMD gene called JAG2, and we collected a number of families from around the world. One of the things that's helped us with gene discovery, in addition to genetic sequencing technologies, is advances in muscle MRI, and these patients had a distinct pattern on their muscle MRI that helped us realize that there is a common pattern that's emerging. And so muscle MRI has become a very important tool for diagnosis and tracking progression, and also for research studies. So for new sequencing technologies, again, looking historically, the karyotype was discovered a long time ago, and that's still useful today in certain situations. And then you'll see a long list of different technologies that have come up since then, some in the 20th century, and then some of them developed in the 21st century. There's still no genetic test method that's perfect, and by perfect I mean by picking up every single genetic variation that's relevant to human disease and not missing anything. So one project that we've been working on for a number of years now is nanopore sequencing, and we are focusing on limb-girdle muscular dystrophy as a target disease category just because there are still so many genetic mysteries. And this just shows the flow chart of how we go about analyzing these patients. My lab and Christine Bruhls and several other people are representing our team here, but Christine has a poster up that has more details of these findings, but we've now sequenced over 61 patients. Since this table was put together, we've sequenced even more, and we found variations that have explained what's been going on with a number of these patients. So among the 61, we've published the first 10. This is back up several years ago now, and so we're hoping that as we gather more cases, we can publish a larger group in the near future. So some of what nanopore sequencing is helping us doing is solving some mysteries that really persist in LGMD genetics, and missing variants being one of the key questions, but also phasing, which I think Lee Kugelman mentioned that during her presentation. When we're talking about a recessive disease and we find two variants, are we sure that they're on two different copies of the gene? Because that's an important thing for linking it to disease. So this is an illustration of how we were able to sort out a beta-sarcoglycan case. And this illustrates, and I won't go into too many details in the interest of time, but this illustrates how we found the two variants, one of which was found commercially, and then confirmed that this was truly what was happening based on some PCR analyses and Sanger sequencing. We've recently discovered more of the power of phasing, which is we can figure out whether the two changes are on different copies of the gene just based on the single sample. Traditionally, you're supposed to get parental samples or other relative samples to really prove that the two copies are on different, that the two changes are on different copies. But now we can do it just from a single DNA sample on the affected individual, and this is for AN05. And then we have other examples for calpain. And the other thing that we are still doing is we're looking for new genes, and this is a gene called myo18B. So this gene's been linked to human disease before, including some potential muscle diseases, but it, to our knowledge, has not been linked to LGMD before. So this is something where we hope that we can find additional cases and see if we can validate this a little bit further. So in conclusion, the discovery of new muscular dystrophy genes always starts with patients, regardless of the techniques used. It's been over 30 years since the first LGMD gene was discovered, and we now have identified 40 or more genes. The new sequencing technologies are helping us, but we have other challenges, including interpretation, as you saw from Chris's talk. And then now more than ever, if you have ongoing dialogue between patients and healthcare professionals, that'll really optimize genetic test interpretation and also help us figure out what's going on. And I appreciate the comments about our study going on downstairs. So Christine and Seth are downstairs. For anybody who has kind of an uncertain result on genetic testing, whether it's a totally unknown case or whether you have part of an answer but not a full answer, please come talk to us. Or if you just want to hear more about what we've been discovering, you're welcome to stop by anytime or talk to me. And so Seth and Christine are in the back row of this photo on the left. Seth is on the left in the back row and Christine is right next to him, and several others in the lab have really been working hard on this project. So we're really grateful for that. And for all of our supporters. And the last comment I'll make is for anyone who likes writing medical articles, Neurology Genetics is a great destination. And so please come also speak to me if you're interested in learning more about that. So thank you very much.